So welcome back, everybody, to the new episode of the Crosse Thinkers Podcast. And today we have Dr. Peter Wilker coming from the Department of Microbiology. And Peter was nominated by one of our listeners to talk about immunology and influenza. So thanks for joining us, Peter.
Yeah, thanks for having me. This is fun.
Cool. So since you were nominated by a student, the first thing he wants to ask is actually, why did you pick this field of study?
Oh, boy. This is really a story of serendipity. I'll give you a little bit of my background. I did an undergraduate degree in biology and never really took advantage of all the different opportunities I had, so wasn't super well positioned to graduate and go out and work in the world. But eventually I found a job and did some work, and that was in Minnesota. And the company I was working for in Minnesota went out of business, as some small biotech companies do, and I started looking for another job. And one of the things that happened at that time was I found a job posting at a place called the Blood Center in Wisconsin. It's in Wauwatosa, Wisconsin. And it was a job posting for a position in a lab of a physician I had met probably about 5 years ago when I was in college. And so when I was in college, I was going to do a study abroad program. And as part of that, I had to go get a physical. And when I went to get my physical, they found out that I had a really, really low platelet count. So platelets are the cells of your blood that are involved with blood clotting. And there's a number of different reasons that you could have a low platelet count, from things that are really benign and not a problem, and all the way up to different types of cancers. And no one could really figure out the cause. And when I was being worked up for that, eventually they sent me, I was living in Minnesota at the time, they sent me to the hospital in Milwaukee because there was a doctor there who was an expert in platelet biology and platelet disorders. And, you know, he treated me as a patient, and then 5 years later I stumbled across a job posting from that same physician. And so I went in, got an interview, and he remembered my case, and I think he hired me only because he remembered me as a patient, because I really didn't have too many skills at that time. But this was a lab that studied immune-mediated blood cell destruction. So sometimes people make antibodies that bind to their own blood cells and destroy them. And so that was really my first introduction to immunology. And I loved it, and I think it's really just because of that weird series of events that I ended up going and pursuing my PhD in immunology. So sometimes life just kind of randomly shows you the way, I guess, right?
As long as you keep your eyes open, and when opportunity comes, you just go right into it.
Yeah, absolutely.
Don't be afraid, and appreciate all the relationships people have and the opportunities people have.
Absolutely, yeah.
Interesting. So let's go a little bit into immunology. Can you give us a brief background on how that immunology works in our body?
Well, sure. Boy, immunology is a pretty complicated subject, and the reason it's complicated is that there's a lot of different parts doing a lot of different things, which is a good thing because bugs are smart too, and microbes tend to figure out a way to interrupt a couple of things, but can't always figure out how to interrupt it all. So we're reasonably well protected. The general way the system works is that you have a series of cells in your body. Probably the most interesting and important ones are the lymphocytes, or your T cells and B cells. And they exist in a number of different forms, but their overall job is to identify anything foreign that gets into your body and eventually attack and destroy it and eliminate it from your body. And so that can be things like viruses or bacteria, protozoa. Lots of different types of pathogens would be recognized by those cells and ultimately killed and cleared from your body.
Cool. So the first thing I know about vaccine as a layman's words is actually, the immunology is actually vaccines from layman's words. So how does vaccine work exactly?
So vaccines are composed of, in some form, some of the foreign materials that would be part of a microorganism that's capable of causing a disease. And so we can make those foreign materials from the microorganism in a lab, and we can administer that to people. And when you inject those foreign materials from the microbe into an individual, those B cells and T cells I was just talking about, they recognize it as foreign, and they respond. And part of that response involves B cells making something called antibody molecules. And the B cells will make those antibodies, and then a special type of B cell forms called the plasma cell. And the plasma cell exists for many, many decades after vaccination, oftentimes, and they just keep making more of that antibody. And it's the job of the antibody to bind to those foreign substances if they should ever get into the body again and destroy it. So the vaccine was made of foreign material from the microbe. You generate antibodies against that foreign material from the microbe. And then later in life, if that actual microbe gets into your body, the antibodies are there already to attack and destroy it.
Will the process work if you somehow inject the vaccine after the virus is in our body?
The only example I know of that is rabies virus infection. So if you are bitten by an animal and you are not sure if it had rabies or not, but there's some reason to be concerned that it did, they will vaccinate you after that potential exposure, and it will provide a benefit. Because without it, if you did contract rabies, it's pretty universally deadly. Mm-hmm. And a lot of that has to do with the specific nature of the rabies virus. It infects neuronal cells, neurons. And if you get bit on an extremity, say on your hand, the virus will get into the neurons down there. And it's really not a problem until the virus moves up the neurons and gets to your brain. And then it's too late. And it takes a while. So if you're exposed, we vaccinate, give the immune system a head start, and then the immune system can usually stop the spread of that rabies virus before it gets to your brain and causes death, generally.
So it's technically still before, not after.
It's before the problem.
Okay, before the problem.
Yeah.
Interesting. So, how do we, so it seems like this is almost like we need to prepare before the disease actually hit us or before the virus actually hit us, right? So
for vaccines, yes.
For vaccines, basically we need to have a big library of all the potential ones which are serious, and then we just do them when we were a kid or do it whenever it's necessary.
Correct.
So how do we predict, for example, especially like the flu shot, do we actually get a vaccine from the flu shot?
Well, we do have a vaccine for flu, and again, the storyline for these vaccines really differs by each particular pathogen that you're talking about. And flu's kind of a tricky one when it comes to vaccination. And the reason for that is vaccinating against flu is like vaccinating against a moving target.
Yep.
Because influenza viruses are able to change pretty rapidly when compared to other pretty common pathogens. And so, you know, the flu virus that was affecting people last year is going to look a little bit different than the flu viruses that are going to affect people this coming season. So it's just a little bit trickier. And so the way we handle that is the World Health Organization conducts a lot of surveillance all around the world. And they have surveillance activities that, in general, are collecting samples from people and from birds and from pigs, and just kind of surveying what flu viruses are out there. What does the most common flu virus of people look like this year? And based on all of that information, when they're getting ready to start production of next year's flu vaccine, they analyze that surveillance data and just make a prediction about which viruses they think are going to circulate the following season. So it really is an educated guess. And sometimes they get it right and sometimes they get it wrong. And when they get it wrong, the flu vaccine isn't as effective as it needs to be. And when they get it right, it's much better and still imperfect. And you hear a lot of people kind of hem and haw about actually receiving the flu vaccine, or they tell their anecdote about getting the vaccine and still getting flu. And a lot of that has some truth to it. But I would argue that even in a year in which the flu vaccine maybe isn't as effective, it's very likely it has some other benefits in terms of perhaps reducing illness severity. Maybe you still get flu, but you're not sick for as long, or it's not as quite a severe infection. So there's still a benefit that sometimes goes unappreciated.
So when you say you predict what's going to be popular for next year in terms of flu virus,
Yeah.
do you mean you predict you have 30 like different ones you have already seen, and then which one is going to be, or actually do you predict how that thing is going to morph?
That's a great question. Right now, we look at the surveillance data and we make our decisions based on what we know to be out there. There has been a lot of work to see if we had the ability to make, in a predictive fashion, better choices, to predict the nature of the evolution of the flu virus in nature before it actually happens. And so, maybe 5 years ago, there was a study that actually came out of the lab I worked in Madison, where in the laboratory, they took flu viruses and basically used the antibodies that people have against flu viruses to direct the evolution of the flu virus to see what it would become in response to the antibody pressure. So the reason flu evolves is to escape the antibodies that exist in the human population. Because if you have antibodies against flu, flu can't infect you, and if flu can't infect a host, it dies out. So flu gets around that by mutating. So they applied this antibody pressure to these viruses and watched how the flu virus evolved. And it turns out that the predictions that came from that study, they had a lot of, they were very accurate with respect to the evolution that actually took place in nature. I don't think we're willing to put all of our eggs in that basket quite yet, because it's still be a little concerning and risky to base all of your flu vaccines globally on that type of an experimental prediction. But people are still thinking about it and still working on it.
At least it's a good aid to our current process, right?
Yes,
It's
absolutely.
ahead of time.
Yeah.
But through this kind of process, do we actually, by injecting antibodies to our body, do we artificially accelerate the evolution of the flu virus?
Oh, by vaccinating and getting antibodies?
Yeah.
That's a fair question. I would instead argue that without vaccination, more people will actually have flu and will generate antibodies because of an actual infection instead of having antibodies as a result of vaccination. So people are going to have those antibodies. It's just you make the decision about if you want to generate those antibodies because you've had an infection and are miserable, or because you have received the flu vaccine.
So basically we reduce the suffering, but basically
Yes.
the process are the same.
Yeah.
Which is being said then, other than you will go through the symptoms, right? Is there any harm for not taking the shot?
Well,
Because you can see a
if
lot of
you're a
people start to
masochist, I suppose not. There's a very serious harm for the very young and the very old, or people who have other medical problems. Flu is something that kills people. Absolutely. I think if you look at seasonal influenza viruses, and average it out, maybe 30,000 people a year die as a result of a flu virus.
globally.
No, in the United States.
Okay,
Sorry,
yep.
absolutely. So it is a serious virus. And some years it's more mild, some years it's a little bit more severe, but it is really a very significant risk to elderly people every year. I've actually never had flu, but it should not be confused with something like the common cold. You know, we've all had the common cold. It's annoying. You feel bad. Maybe you miss school, maybe you miss work, but flu will knock you on your butt. It's a whole different level of an infection. And so I got my flu vaccine last week, actually. And I try to do it every year because I just don't see a downside to it.
So, taking your words on that, is it fair for me to say taking the injection vaccine does not have harmful side effects on a person's body?
the frequency of severe adverse reactions to the flu vaccine is very, very low.
What do you mean by adverse?
So every medical procedure, of which vaccination is one, carries some risk. So no one will ever tell you it is perfectly safe. But very few people have problems with it. Very, very few. So the benefit by far outweighs the very infrequent problem that we experience. Most people, when they talk about problems with the flu vaccine, you have some soreness in your arm for a day or two. The flu vaccine does not cause flu. The virus material that's in the flu vaccine that gets shot into your muscle is absolutely not capable of causing infection. It's been treated through production to kill the virus. It actually goes through rounds of making sure that virus is killed. And then the virus particle is split into pieces. It can cause you no harm. But yeah, you might have some soreness in the arm.
Okay. So, I don't know anything about this, but I have a question which bothers me a lot is, why flu? Like, there are so many diseases. If once we develop the antibodies, or if we develop all those kind of vaccines, they basically went into extinction. But why flu just keep evolving? Is it because...
There's a lot of different kinds of viruses. And each different kind of virus works in a very different way. So with respect to flu has a genome, like all viruses do, but its genome is made up of RNA. Your cells have, as a genome, DNA. And there are other viruses like smallpox, since you mentioned disease eradication. Smallpox had a DNA vaccine. And the importance of that really has to do with how quickly they mutate and change. When you copy DNA molecules, that's copied reasonably well, so there are very few mistakes. When RNA is copied, as has to happen for an RNA virus like influenza, it makes lots of mistakes. And those mutations cause genetic diversity within the population of viruses. And genetic diversity is really the starting material for all evolution. And so for that reason, influenza viruses have the capacity to mutate, evolve, and adapt to new conditions much more rapidly than a lot of other pathogens out there. The other thing that's a little bit special about flu is, there's a bunch of different flu viruses out there. Only a very limited number of them can infect humans. And they all work in generally the same way, but some of the influenza viral proteins seem to have really extraordinary flexibility. They can mutate, change their overall shape a little bit, but still retain the overall required function of that protein as it relates to the influenza viral life cycle. It can still get the job done. But that kind of changing of structural shape of those proteins is what really allows humans to be able to be reinfected over the course of their lifetime, because it has implications for whether or not the antibodies you've made in the past can or cannot recognize the altered, evolved, and adapted virus.
Interesting. So are there any other virus like this other than influenza?
There are. HIV is a great example. In fact, the extraordinary genetic diversity of HIV that exists across the world is the main reason we don't have an HIV vaccine. People have worked on an HIV vaccine since the virus was discovered in 1983. And it's not for lack of trying that we don't have a vaccine. It's simply that HIV has mutated so quickly and evolved, and there's so many different types of HIV that we don't know how to vaccinate to protect you from all of them. HIV is a little bit of a different virus as well because it establishes lifelong infection in people. Flu comes, you get sick, your body responds and clears the virus. For HIV, if you're infected, it's a lifelong infection. You never get rid of the virus.
Why is that?
It has to do with the fact that it's a virus type called a retrovirus. And so what happens with HIV is it takes an RNA genome and it makes a DNA copy of it, And it actually takes a DNA copy of the virus genome and literally sticks it into the genome of your own infected cells. So all the genetic material needed to make more HIV viruses is encoded for in the DNA of an infected cell. And so if you were going to hypothetically cure somebody of HIV, you'd have to do two things simultaneously. And right now we're nowhere near being able to do that. You'd have to get rid of every HIV particle in the body, and you'd have to get rid of every cell that's harboring the genetic material of HIV at one moment in time. And we don't know how to do that.
So for the first challenge, we can probably do it with antibody, right? Eventually, to remove all the particles of.
Well, even that's going to be a problem.
Why is that?
And it's a problem that's very similar to what we were discussing for influenza, which is as HIV is making more viral particles, it makes a lot of mistakes. And so for flu evolution takes place quickly, but really there we're talking on the order of years when we're talking about seasonal influenza virus. HIV virus evolves in the context of one patient.
That's crazy.
And you can watch it. So there have been studies that have collected samples of virus from a single infected patient, and you can watch in real time how that viral population diversifies over months or a year. It's really remarkable. And again, as the virus mutates, it's changing just subtly some of the structures of the viral proteins so that the antibodies that were made to combat HIV no longer are as effective. And so the immune response has to basically start all over in that patient. Now we got to target those changed HIV particles. But then the virus changes again. And the biggest problem with HIV is that the HIV virus actually replicates in one of these types of lymphocytes that we were talking about. It's called the T helper cell. And that's one of the cornerstones of all of the immune response. You can't really make antibody without T helper cells. Nothing works as well. And that's the cellular target of HIV infection. And so over time in HIV-infected individuals, their CD4 T helper cells start to disappear. And eventually they go so low, they just don't have enough T helper cells that their whole immune system collapses. And then usually what actually ends up killing those people is they get infected with some other microorganism because they can't defend themselves against it.
So that's the common hearing I got from HIV destroy your immune system. So that's actually exactly how.
Absolutely right.
Oh, okay.
Yeah. So really it's really an unfortunate series of biologies for that virus that have kind of coalesced to make it a really horrible problem. Antigenic diversity, you can't get rid of it from your body. In terms of the epidemiology, you're infected, lifelong infection, but you might have HIV infection for 5, 6, 7 years and not feel sick at all. So you might not know you're infected, in which case you might be engaging in activities that are spreading it to other people. That's a big part of why it's such a big problem globally. 37 million people infected worldwide. It's really terrible.
Next generation too, right? If you become pregnant during this process, the baby is going to have to carry that too.
Well, that's exactly right. So you can think about this in countries that have a high HIV prevalence. So some countries in the southern part of the continent of Africa have adult HIV prevalence rates that are 20%. So 1 out of 5 adults is HIV positive. And so you're exactly right. In those countries, if women are getting pregnant and having babies without any type of drug intervention, those babies can be born HIV positive. The good news is, well, at least partially good news, is that we have a lot of good drugs to manage HIV infection. And so people who are on those drugs can live pretty much normal lifespans at this point. And the other component part of that is if you're on those antiretroviral drugs for HIV and taking them as you should be, you will not transmit to another person. If you're a mother, you will not transmit to your developing fetus or newborn. So it both is treatment that is treating the patients and reducing misery, and it is a treatment that is preventing transmission events and new infections.
This treatment, what's the cost? Like, is it affordable? Because it's
That is the biggest problem worldwide. And so the cost is progressively getting better. I don't know exactly what it is, and it differs country by country, actually.
Do you have like a rough number?
I would hesitate to even
Thousands,
guess because
hundreds?
I would probably be wrong.
Okay.
I think it can be made available in poorer countries for dollars a day. Okay. But that's a drug that people need to be on every day for the rest of their life.
Yep.
And it's an actually terrible situation if you start somebody on these drugs and then they start taking them sporadically, because HIV can mutate and maybe develop some level of resistance to these drugs. And that would be a disastrous situation. So the affordability of drugs, the availability of drugs is still a major limitation to getting people on treatment worldwide. Absolutely. There's a lot of work being done to change that, and there's a lot of progress being made, and that progress is kind of starting to have an effect in terms of the fact that we're starting to see lower incidence, new infections of HIV in a lot of these countries that have historically had really massive problems.
Just listening to your description, this almost feels like an ultimate biological enemy for our body, because it targets every single thing we do to get rid of.
No, I agree. I've thought about it the same way. It's like, wow, what a clever. Obviously, it's not clever, but if it could be clever, this would be one of the most clever viruses.
Feels like a design, right?
Yeah.
Another thing is just, thank God, like it's only spreading a certain way, so at least we can put control on that. If it's spread as influenza, basically we're done for.
That's true. HIV is actually not that easy to spread to another person, but it's spread by behaviors that people do pretty frequently. So sexual transmission.
By natural selection, why don't they just, take over the world? Is it because they're going to kill every single?
Well, it's not in the best interest of microorganisms necessarily to kill their host, right? The only circumstance where a microorganism really wants to kill their host is if it allows them to more easily be transmitted to the next host. So it's all about having opportunity to create progeny, new viruses, new bacteria, whatever organism you want to talk about. And so generally, for a lot of these different pathogens, you can think about flu again, the signs and symptoms that you experience are actually the same thing that facilitates spread of that microorganism to the next host. So for flu, well, I get a runny nose, I get a cough, I might sneeze. If I cough and I sneeze, I'm coughing and sneezing out viruses into aerosols that are going to be breathed in by another person in the room, and that's going to be a transmission event. The virus lives on. So, I think there's a limit to how much damage these pathogens really want to inflict on a host. Because if a pathogen causes quick, immediate death, well, that might come at the expense of the pathogen spreading to another host, and then it would die out.
Makes sense. So it's more like they're trying to go into a coexisting situation
with
their host, and it's just good for them and bad for us.
Yes.
Interesting, yeah. So we talk about vaccines, which use the system of our immunologies to protect us from virus or potential diseases. Are there any other kind of therapies we can take with the idea of immunology other than vaccine?
Oh, well, if you have a bacterial, not prophylactically or for preventative purposes, I'm not coming up with much.
What about therapy? Because I saw a terminology here called immunotherapy.
Yeah, we can talk about that. Well, let me stick on this thread just for a second. I think it's important to recognize that for bacterial infections, we generally have very good therapies, just antibiotics. For viral infections, That's a little bit tougher. There, for some viruses, are some antivirals, but in general, they're not that great. So there are some therapies, at least for bacterial pathogens, for a lot of fungal pathogens, for some protozoan pathogens. So there are other ways that we can deal with those infections if we become sick. Immunotherapy is really, really a neat subject. It takes us away a little bit from our discussion of infectious disease, and probably moves us a little closer to talking about things like autoimmunity and cancer therapies. But right now, the field is really exploding. So what we're talking about with immunotherapies are really making, in a laboratory environment, protein molecules that are either going to inhibit or promote some aspect of the immune system to achieve an outcome. So I'll give you an example of that. In an autoimmune disease, a person has lymphocytes, T cells or B cells or both, that mistakenly start attacking self. So most people don't have that problem. Your immune system doesn't attack your own body. But in an autoimmune disease, something's gone wrong, and you have autoimmune attack. So what would we want to do? Boy, we'd really like to slow that immune response down. We'd like to put the brakes on it. And so what immunotherapy does is, well, it all starts with basic research. We'll study an autoimmune disease. We'll try to better understand how the immune system is working to cause some type of tissue damage that is the autoimmune disease. We'll look at the players, what cells are involved, what proteins are involved. There's some really important proteins that are generated by immune cells called cytokines. Sometimes certain cytokines cause some of the damage. And once we know enough about an infectious disease process, just how does it work, then we can start picking out some targets. So in some diseases, there's a cytokine called tumor necrosis factor alpha, TNF alpha. And that cytokine seems to drive some of the tissue damage in autoimmune diseases. And so what we've done in terms of immunotherapy is we've made products that recognize that cytokine that does the damage and neutralizes it. So in some cases, the immunotherapeutic drug is actually an antibody molecule. So in a lab, we make an antibody that binds to and recognizes that TNF alpha cytokine that does the damage. And when it binds to it, it neutralizes it so it cannot have an effect. So that's one type of immunotherapeutic approach. And there are antibodies against lots of different targets for lots of different purposes. I can give you another example related to cancer. So there are some B-cell cancers. So cancer is when a cell loses control of its cell cycle and just starts dividing and making more of itself and more of itself and more of itself. It just keeps going. And so some drugs for these B-cell cancers are antibody drugs. And what we know about B cells is B cells have a particular protein that's expressed on their surface. It's called CD20. And that protein, it's expressed by B cells, but not by any other cell of the body. And so if we have a cancer that's derived from a B cell, that cancer cell also expresses that protein, CD20. So the therapy that's been developed is an antibody that binds to that protein CD20. Since that molecule is on the cancer cells, the antibody binds to the cancer cells and flags them for destruction. Then the immune system comes in and kills the cancer cells. Of course, normal B cells also express that protein, so at the same time we're going to kill off our normal B cells. But we take care of the biggest problem first, which is the cancer. And we can help people along until they're able to make more of their own B cells, assuming the therapy is successful and we get rid of the cancer. So that's another example of where these immunotherapeutics have come into place.
So it's almost like you build antibodies, but this time the target is not virus coming from outside, but it's more about coming from your immune system yourself.
Yeah.
And then you just go there and use your own army to basically control the army of another state.
Yeah. So those are two examples where we're making antibodies in the lab and then administering the antibody to people for therapeutic effects. There are other classes of these biologic drugs, these immunotherapeutics, that are intended to, in some cases, stimulate a person's own immune system such that it mounts a more vigorous response against things like cancer. So it might be a little bit counterintuitive, but the immune system is actually very, very important in defending the body against cancer. I say it might be counterintuitive because, well, cancer cells are self cells generally, right? They're cells that are doing something that is undesirable, but they're built of pieces that are just like the pieces of all the other normal healthy cells of our body. And so generally our immune system doesn't attack self. But there are enough subtle differences with those cancer cells that gives T cells and B cells of your immune system some targets to recognize. And so some of these therapeutics that are being developed are basically intended to take the brakes off the T cells and B cells that are responding to the cancer cells. The way normal T cells and B cells work against infection is they'll mount a vigorous response and then the brakes get turned on when the threat goes away and everything kind of settles down. But with cancer, we want to keep the brakes off. I want those T cells just to mount as vigorous a responses as possible to make sure I get rid of every last one of those cancer cells. And so some of these drugs are targeting those brakes.
Interesting. So it provides a possible way to cure cancer as long as you can target a certain type of cancer cell and basically pass this target to the immune system and tell them, hey, when you see this, don't stop.
Yeah,
just
so
go ahead and clean it.
Right, that's true of. So sometimes we need a dedicated target that we know is on the cancer cell.
Mm-hmm.
These drugs that are just taking the brakes off a normal immune response are really intended to just let your immune system do the work it normally does, which is find something different, find something that looks like it shouldn't be there, attack and kill it. So in those cases, we maybe don't even have a specific molecular target defined for the cancer cell, but we let the beauty of the immune system do that work for us. We just kind of unleash it using these drugs.
So that's even better. You don't have to design.
Yeah, because it's actually pretty rare that we have a great target on cancers.
That's actually my next question.
Yeah.
Because when I was thinking about target, then that will be limited to some cancers that has a significant feature about the cell. But how universally applicable for those kind of immune knowledge therapy for cancers?
It's starting to be a little bit more widely applied. To the best of my knowledge, it's mostly being applied to blood cell cancers. And that's generally because we can identify good targets. And in the example I gave you, my example was, well, we'll kill the cancer cells, but we're also going to kill the healthy B cells that gave rise to the cancer cells. With a lot of our T cell cancers, B cell cancers, we can actually live for a period of time without the normal T cells or B cells, because there's always going to be a little bit of collateral damage. It is not the case that we generally have a marker that is fundamentally restricted to the cancer cells. It's usually a marker that's on the cancer cell, but is also on some other normal healthy cell. And most of these therapies are going to do damage to those cells as well. But with respect to T cells and B cells, they're not essential for survival today. We can help people along and fill the gaps of not having B cells to keep them alive from an immunological perspective while we treat the cancer.
Big problem first.
Big problems first, yeah.
So is it fair for me to say this kind of modifying the behavior of your immunology system is temporary, or actually is there a way you can just permanently tweak it and then say from now on, even if you stop taking a certain kind of thing to trigger that, this thing will behave from now on?
Well, so in the cases we've been talking about, we were talking about administering antibodies. Mm-hmm. to mediate that effect, to either take the brakes off or to attack a cancer cell. And antibodies are proteins, and proteins have a half-life in the body. So unless you keep administering those, eventually the drug goes away. Okay. There's another type of immunotherapy, and this one's a little different. It's a little more complicated, but it's really, really amazing and interesting. They're called chimeric antigen receptor T cells. Sometimes people just call them CAR T cells. And so this is a cancer therapy. And the general idea here is you have a patient with a cancer, and this would be a cancer very much like the one I talked before. So let's consider the case of that B-cell cancer with that molecule CD20 that's on those cancerous cells. What they do with these patients is they're actually going to take some blood and from the blood purify their T cells in a laboratory. And in the laboratory with those T cells, what they do is they use a whole bunch of tricks to actually engineer those T cells to give them a very different type of receptor that's not made naturally by your body ever. It's kind of Dr. Frankenstein work, weaving things together to make a receptor that can do something different. And they give that extra DNA encoding that new receptor to those T cells, and then return those T cells to the patient's body. And what that new receptor that the T cells now have what it allows the T cells to do is to recognize that CD20 molecule on the cancer cells. And then one thing that some T cells are able to do when they have this recognition event is they secrete some substances from the cell that causes death of the target. So it's a genetic approach that actually reprograms a patient's T cells, which are returned to the body, and then, because they're reprogrammed, very efficiently seek and destroy the cancer cells. But you're right. Maybe we don't want those cells to be around for the rest of the patient's life. And in fact, sometimes those...
Without keep taking, antibodies or to keep taking medicines, right?
Yeah. Because they're cells and they can persist for a long time. Yeah. And in that case, those cells are going to respond to CD20 on the normal B cells even after the cancer's gone. So even after the cancer might be cleared, if those cells were allowed to persist, they would be killing normal healthy B cells and the patient would be immunodeficient for the rest of their life. So researchers are clever. And so this has gone through a series of generations of designs now. But now what they do is into those T cells that they're modifying, before they put them back into the patient, they engineer into it what's called a kill switch. And essentially, it's a switch that's activated by the administration of a drug that causes those engineered T cells to die. So we engineer the T cells, engineer the kill switch, put them back into the patient. Those engineered T cells kill the cancer. Once you're convinced the cancer is gone, you administer another drug that activates the kill switch, so those modified T cells die off.
It's amazing.
Yeah, it's really, really amazingly clever stuff.
It's building small robots in your body, and you have a switch to actually kill them. It's just they're not electronic or metallic.
And the beauty of it all, I teach this in my immunology class. It's one of my last lectures of the year. The beauty of that is you see how the basic science is taken and then applied in such clever ways to make these amazing therapies. And these therapies that we've been talking about, they just won, some of the key scientists who developed them were just awarded the Nobel Prize last year. So it's really, really important stuff with a lot of promise. It's really expensive stuff. So those that T cell therapy, I think, is $300,000 to $500,000. So we have some things to work out in terms of insurance and how much access people will have to these type of therapies as they do become more common and are shown to be really, really good therapeutic options for people after many of the other more common chemotherapeutical interventions have failed. But we'll see where it goes. It's neat stuff, though.
It's really interesting, yeah. With all the basic ideas putting together with some simple mechanisms, the idea totally makes sense. It's just like when you implement it, all kinds of stuff just pop up, and you do it in a really efficient and correct way, right?
Yeah. Let me mention this to you, because this is kind of another neat aspect of those T cells that I was talking about.
Mm-hmm.
So I say they reprogram the T cells by adding some DNA to those patient T cells. And actually, the whole system for delivering that extra material is based on the biology of HIV. So we've taken HIV, this horrible virus that's caused tens of millions of deaths worldwide, and we engineer that to be used for some therapeutic good. So even there have been a number of tools like that have actually come from our discovery of HIV. Still a horrible virus, but there are some things that come from our research of that have been applied elsewhere in really interesting and important ways.
It's beautiful.
Yeah.
It's basically we're training HIV as an animal, as a farm animal now.
Yeah,
right?
we're putting
only
it to
take your meat and whatever toxic we throw it away, but
absolutely, we're
it
putting it to
definitely
work
teaches
for us. Yeah.
And what about transplanting of organs? Like with this kind of thing, sounds like you can basically put a break on your immune systems whenever you have a conflict with.
Transplantation biology is really extraordinarily difficult. So the difficulty with any type of transplant has to do with the fact that if I take an organ from one person and I put it in a new environment, well, you're different than me. You're genetically different than me. And that means the tissues that you have don't look like the tissues that I have from an immunological perspective. So the whole basis of the immune system is to recognize things that are foreign to yourself and attack and destroy it. And so most organs are going to be recognized in some way, shape, or form as foreign to the individual that received it. And there are a few different processes that account for immune system rejection of a transplanted organ. And we know quite a bit about that now. So you've probably heard about matching, finding a good match. So one thing we
reduce
do
the differences, basically,
reduce the most important differences.
Okay.
So if we reduce the most important differences, well, unless you have an identical twin, there's still differences, but they're a little less important. And then we can handle the immune system response a little bit more easily with drugs. And usually we use some type of an immunosuppressive drug. Sometimes there are those therapies, based on the therapies that we were talking about a little bit earlier, more often they're small chemical drugs that inhibit the immune response, just to slow it down. And by matching, we've just restricted the most important differences enough to give us some hope that with drugs, we can slow the immune response down so that organ that's been transplanted survives and functions long term.
What about, actually, I have this confusion. Which is better in the term of like immune system consistency? Artificial organ or the organ from a different person?
Well, I don't think we're to the point where we have options for artificial organs very much.
Okay.
This is a little bit outside my realm. If you could get there, I think that would be great. And people certainly are exploring how to use things like stem cells and scaffolds to generate organs in a laboratory that might someday be suitable for transplantation. But right now, in general, your best option is going to be from usually a recently deceased donor, with the exception of kidney transplants, which can come from a living donor.
Okay, so
there's a lot of work that also is done on maybe someday using other animals for transplantation into humans. So like
Why is that better
pig.
Than from like human to human?
Well, there's a shortage of available organs in humans, right? Because you have to essentially wait for people to have some kind of unfortunate event that leads to their death. And then even more difficult is now we have to find the best match. And so there's a lot of people that die waiting on the transplant waiting list. So if someday we could get the science to the point where we could engineer and raise pigs, you might have a more sustainable, available source of organs for human transplantation. And there's progress being made, but that's still an awful long way off.
And they will be much harder to match, right?
Well, they would. Well,
organs.
we're clever. Scientists are clever, and so the idea would be to genetically engineer the pig, so that they didn't have those things that cause such a substantial mismatch. So they basically would remove those things from the pig.
So basically, we're talking about the things in sci-fi movies.
Oh yeah,
with our clones and just
you really are.
Organ farms.
Yeah, there's a lot of genetic engineering that's going on out there, and it sounds scary. And if misapplied, of course, maybe it is scary. But I think most of us are well intentioned.
Yeah, the first time I saw this, that makes total sense. It's just as long as you ignore the human rights of the clone people.
Yes.
But
well,
then
yeah, that was
what about
a
halfway,
year
just
ago.
like
Yeah.
I clone some person, but I don't clone them completely. Only the top half, no brain, no anything, but they do have the organ I want. And then you're really pushing the line. It's just like, up to which point
well,
this become
I mean,
non-ethical?
Yeah, I think we're already to a point in science where the question isn't, can we do this? It's becoming increasingly, should we do this? And that's not a new story. That's been going on for decades. With every new technological advancement in science, it raises the should we question. So I don't think that's new, and I don't think it's going away.
Are there any big ones in immunology that you shouldn't be doing?
Well, I've got a good should we for flu. So it has to do with avian influenza, bird flu. So bird flu viruses infect birds, and there's a couple types of them that very sporadically and infrequently infect human beings. The bad news is that when human beings get infected with these couple types of bird flu viruses, it's lethal about 50% of the time. So very, very severe. When we talk about seasonal influenza virus, the death rate is less than 0.1%, and usually they're very old and very young. Here we're talking about 50% of people who are infected die. But the good news is when a person gets infected with bird flu, they don't transmit that virus to another person. So it's a virus that sporadically infects people, frequently kills people, but is not transmitted between people. That's the good news.
How did they get infected from the beginning, from the birds?
Oh, it usually requires very close contact with poultry. So poultry farmers in China takes a lot of contact.
So it's those kind of physical contact and close.
Yeah, living in close proximity, butchering poultry, blood, right? All that, I think, is what is required.
I see.
And so a question for influenza virologists is, could these viruses become human-to-human transmissible? Is it possible? Because if it is, that's a little terrifying.
Yeah.
Right now you have a flu virus that transmits through respiratory droplets, person to person, and maybe kills people 50% of the time. That would be a disaster. And so there's been work that's been done in the laboratory. People have started with these bird flu viruses that are not transmissible, and they've manipulated them in the lab. And through a series of experiments, they demonstrated that, okay, if I change this bird flu virus in these 3 or 4 ways, now it's a virus that in ferrets is transmissible. And ferrets are the model used to demonstrate person-to-person transmission. They cough, they sneeze, they transmit just like people do. And so those bird flu viruses and the information related to those bird flu viruses is out there. They're in labs, but transmissible avian influenza viruses exist in a lab. They don't exist in nature. So the question is, should we have made those, right? People have laboratory accidents. They're handled in very, very safe environments in these biocontainment labs with trained staff. but is anything perfect? And what would be the outcome if one of those viruses got out of the lab and started to spread through a community, right? So that's one more modern day example of should we? And the government actually put a pause on all of that kind of work for a long time while they thought about the should we question. And so now there's just systems in place about conducting that kind of work, proposing that kind of work. You basically have to pitch it to people who review it in advance.
So it's almost you're creating natural biological weapons, which would target people's in terms of like a disease, which is hard to recognize.
I mean, I think all it takes is one person with a little bit of a scientific background and experience in microbiology to do something like that.
And curiosity.
Oh yeah,
or
you gotta
some
be
other
a
intentions,
little. You gotta have a little bit of a. You gotta either be angry or have some type of a mental illness. But people do get angry and people do have mental illnesses, and I think that's actually a big concern about the people who do work with these really much more dangerous viruses out there.
So it's the 12 monkeys.
Yeah, or the 2001 anthrax attack. Right? He had a government employee. We don't really know why, but he got his hands on anthrax that was produced by the government for weapons research, presumably, and sent it through the mail as a terrorist attack. So, most people don't think like that. Very few do, but it's a concern and something that people should spend time thinking about.
Yeah, I kind of feel like our advancing science is right at the stage of we can basically have millions of ways to kill all the human beings on the planet, no matter it's physics or academics or chemical or whatever, right? Nuclear weapons, chemical weapons, all these kind of things.
Yeah,
and it's engineered to such a precision that it's efficient and quick. And hard to defeat because they target right at your weakness.
Yeah.
Right?
Well, and that's one of the reasons that biological weapons, I think, were pursued is, well, one, if I make a biological weapon, I can make the weapon and I could probably make the vaccine to protect myself and whoever I want to protect.
You can target your enemy genetically.
Right.
Yeah.
And so I think it's largely thought that part of the reason that the Soviet Union wanted to eradicate smallpox is so they could develop smallpox as a biological weapon. So let's get rid of it in natural circulation on the planet and then create it in our labs as a biological weapon in case we ever want to use it.
No antibodies anymore. No, it's just like overnight.
Yeah, eventually if it's eradicated, people don't get vaccinated anymore, right? We're all susceptible to smallpox if it were ever to show up again.
It's cheap too compared to other methods.
Yeah.
It's cheaper. Have you ever played
and it spreads.
Have you ever played a video game called Plague?
No, I've heard of it, though.
You heard about this, right? Oh, it's just like the first thing you want to do is actually don't develop any symptoms until 10 years later. So just make sure everybody got infected, and then suddenly overnight, everybody got a fever, and then
and that was it.
Within 2 months or something like that, everybody on the planet dies. Yes. So let's talk about this a little bit. Without vaccine, or just like how deadly those kind of epidemic disease can wipe human beings. How dangerous are they if there is ever an explosion of epidemic hazard? And let's go with the one which is in 1918 for the Spanish influenza. And
we have lots of historical examples of epidemics and pandemics that were quite severe. I mean, you could talk about the plague, bubonic plague. People usually say, 30% of Europe was killed. Well, that probably wouldn't happen again. And, plagues caused by a bacteria. If people got plague, we could treat them. Spread by fleas on rodents. We could control the rodent population. So we know how it is spread, and we could easily intervene, unlike the 1300s. Influenza, in 1918 and 1919, there was a pandemic. So pandemics arise when Mother Nature creates a totally new strain of flu that happens to infect people and spread between people. And in 1918, a strain emerged, probably from birds. And it very rapidly spread around the world. And in less than a year, most estimates would suggest maybe 40 million people worldwide were dead. About 500,000 in the United States alone. And that's in less than a year, right? And just for reference, HIV since 1981 has claimed maybe 35 million people.
Mm-hmm.
Okay, so here we're talking about
within a year.
The entire HIV epidemic over the course of almost 40 years compared to flu in 1 year.
Back then, we don't even have airplane or anything like the transportation, right?
Yeah. Well, we did have World War I.
Yep.
And so we did have troops being moved around the world to spread that virus.
I see.
And that was important. But there were more deaths in the military from influenza than there were from battle. It's not even close. And so that tells us just how severe influenza could be. That was a virus that had a mortality rate of maybe 2.5 to 3%, which actually doesn't sound that deadly. For bird flu, it's 50%. For seasonal virus, it's 0.1%. But so many people became infected worldwide, 1/3 of the global population of the time, that when so many people are infected, even a 2 to 3% mortality rate gets you to a pretty high number, to that 40, 50 million range. It must have been a terrifying thing to live through. At the time, influenza was just a disease. They didn't know what the cause was. They pretty much understood it was infectious, but virology was pretty much a brand new field, just getting started. So there weren't flu viruses, there was just flu the disease. And if you don't know what causes it, boy, your hands are kind of tied to do anything about it.
Do they know how that thing got spread, at least? Do they at least know how that thing got transmitted from person to person?
Well, it's definitely respiratory.
Okay.
Yeah.
So they know that.
Yeah.
At least. Okay.
Yeah. So interestingly, back to our should we conversation, right? So the 1918 virus that was very, very lethal and caused this probably one of the worst pandemics in human history, that virus evolved and disappeared before we knew how to grow influenza viruses in the lab. So it didn't exist anymore. It changed enough through circulating through people that the virus that was so deadly was gone, right? But scientists asked the question, boy, I wonder what it was about that virus that was so deadly. Maybe we could learn something by studying that virus, understanding why it was so deadly, in order to be better prepared for another pandemic. But the virus wasn't around, because that was 100 years ago. But then we make these new technological advancements. And the technological advancement that came along allowed us to be able to basically recreate the 1918 strain of influenza, to resurrect it. So it's totally gone from the world. We had a technological advancement, and we were able to magically recreate that virus. Now that virus exists in labs. I've actually studied that virus.
How did they do that? They take the modern influenza virus and start to mutate it until it happened?
Well, here's how they do it. So the technology that became available basically meant that if you had the genomic sequence of a flu virus, you could recreate the actual virus. All you need is the genetic sequence.
How did they get the genetic sequence then?
So they did it in two ways. One way actually involved opening up a mass grave in an Inuit village in Alaska. It's a little place called Brevig Mission. And so this was a village in 1918. Flu came in and killed almost everyone in the village. And so at the time, people were dying so fast, they'd hire territorial gold miners to come in and dig mass graves. And so it's Alaska, it's cold, and the ground there, it's permafrost. which means the ground never fully thaws. So they had these bodies in these mass graves in the permafrost. And what people did is, after the technology to do this was developed, they asked the village elders in Brevig Mission if they would allow them to open up the mass grave and see if there were suitable tissue samples that could be brought back to the lab. And so they did just that. And from those tissue samples, they were able to collect really hard work, but eventually sew together the entire genomic sequence of the 1918 influenza virus. So that was one approach. The other approach involved archive samples, archive tissue samples that were collected from military personnel during World War I. So many of them died from influenza. And so they had archived medical history on these patients. They'd go through the medical history, find medical histories of soldiers that looked like they had died from something consistent with influenza, and then saw if they had archived tissue samples. And those tissue samples were used to recover genetic information on the flu virus from 1918 as well. And then you get that sequence information, you plug it into that technology, and you're able to recreate the virus for further study.
When were these done, these two approaches?
Oh, the paper reporting that was published in 2005.
So
that's almost 15 years ago.
Yeah, but why did they initiate this thing from the beginning, applying for grants, just because they are curious?
Well,
or
I would imagine if you ask the people who do the work, the argument is because by studying something that was so deadly, we can better understand what it takes to make a flu virus that's so deadly. And in our surveillance activities worldwide, we can look for those types of features in the viruses that we collect and analyze. There is some truth to that. And then part of the truth is probably can do. I'm doing it because I can. And because how crazy would it be to recreate a virus that was gone from this planet that caused the worst pandemic ever known?
It's interesting.
Yeah.
In a way.
Yeah. And I'm kind of in the middle area. I can't fault people. I spent a lot of my postdoc studying the 1918 flu virus, and I studied these bird flu viruses that were engineered to perhaps be transmissible between people. And so I think there are things that we can learn that are important. For example, I think it is important to recognize that the bird flu virus has the theoretical capacity to become transmissible between people. That's an important thing to understand because it informs our risk assessment. It informs perhaps our need to create vaccines in advance of a problem and have them stockpiled. So I do think there is some truth to that, but there is absolutely a risk to doing that kind of work as well.
Interesting. So is the one in 1918 the last severe one, like worldwide?
There have been others. It's by far the most severe. Pandemics are never good for a variety of reasons. The most recent one was in 2009 with what's been called swine flu. And so that was a novel virus that emerged from, as you would guess, swine and infects people.
We better just call it
a day.
I would like to continue the conversation later. Okay. But that was super interesting. Thank you.